3.
Hello learners, this is Dr. Tillahan, and I'm going to explain to you the laboratory today involving the basics of soils and involving biological organisms. And today we're going to extract what are called nematodes from soil. Okay, so nematodes are microscopic worm-like organisms in the soil that are either parasitic or non-parasitic, and they can be endoparasitic or semi-endoparasitic or ectoparasitic. And these nematodes attack roots of plants generally if they're parasitic. The non-parasitic organisms or nematodes live in the soil, feeding off fungi and other bacteria to survive. But some don't feed at all, and they just there for copulation or reproduction, and then they die off. So nematodes are what we're going to extract from the soil today. And so right now I've got my lab coat, and I have my gloves ready to go, and we're going to go ahead and do the first thing. I'm going to explain to you what we need for all the materials and methods of this procedure. And so the procedure involves, first of all, extracting soil from outside of the agricultural building, the School of Agriculture and Applied Sciences, the same area as we used for the mussel colored kit to determine the hue. Okay, so now we're gonna go to the same area. We're gonna use an auger, and that's how I'm gonna push into the soil and collect some soil samples. And we're gonna put the soil samples in a 500 cc or 500 mil or 500 cubic centimeter beaker. Okay, I'm gonna just use a portion of this beaker, but most likely I'll use about 200 cc, 200 mils of soil to extract the nematodes from. Okay, and once we get here, we're gonna use what's called a mesh sieve. This one is a larger grade of a mesh sieve, and it's 850 micrometer diameter here. And so this is a 20 mesh sieve. You can see it's a large mesh here. Mesh, this is a sieve like you sieve flour, okay? And then it's gonna be sitting on top of, like that, like so, of a smaller mesh sieve, the diameter being 38 micrometers, and so it's much smaller. And so this is where the nematodes will be caught. This is where the soil will be watered, will be cleaned out of, and larger particles like leaves, roots, and so on will be caught on there. And then the nematodes will be caught in smaller soil particles in this small sieve. Okay, and then we're going to transfer this portion of the soil into 50 mil tubes, centrifuge tubes. Okay, and we'll pour them into the centrifuge tube, where we then will carry out centrifugation. Centrifugation occurs over here. Come join me in this tabletop centrifuge here. Okay, this centrifuge will go up to 14,000 RPM or 14,000 RPM, or if you want, you can go to gravity. Let's see the speed here. You can set the speed up to 12,000 or 10,000 gravity, but we're using RPM, so it's 14,000 RPM. But actually, what we're setting it at is 3,000 3,000 RPM. Okay, so that's the maximum RPM we're going to use this centrifuge in for. Okay, the centrifuge will be able to contain, if you look in there, it will be able to contain those centrifuge tubes, the centrifuge tubes that I showed you with the nematode samples in them. And then it will spin at 3,000 RPM, and the heavier material will be collected at the bottom, and the lighter material will be collected in the supernatant. And so that will separate initially. Initially, the nematodes will be in the heavier particles in the soil rather than the water. But then after that, what we're going to do is, after we pour off the supernatant, we're going to collect, we're going to pour a 10% sucrose solution into the pellet and shake it real hard, so where the soil is broken up into the 10% sucrose solution. And the sucrose solution has to then go through the mesh sieves again and get washed off as quickly as possible, and in order to allow the nematodes to live and survive the sucrose, because of osmosis. Okay, osmosis is where higher concentrations on the outside of the nematode will pull out the solution, or their inner, like water in their bodies outside, because there's higher concentrations of sucrose outside than there is on the inside of the nematode. So they actually implode. They actually die by implosion instead of explosion, where if they had higher concentrations of sugar inside their bodies and there was no concentration of sugar outside, the sugar would leave their bodies. They would then the water would actually enter their bodies, so their bodies would explode. Okay, but in this case, we're using osmosis, and to where we're going to wash the sugar off so they don't implode. Okay, and the nematodes will live. So I'm going to show you over here where we're going to measure. Where we're going to measure a solution of 100 milliliters of water. In 100 milliliters of water, we're going to make a 10% sucrose solution. And so I've placed this this on a scale, and I've tiered it. And then I measure out 10 grams of sucrose. Sucrose is nothing but sugar. Okay, so sucrose is generally sugar that we use like in coffee or anything like that. I weigh out 10 grams of sugar. Okay, so you see that 10 grams of sugar are weighed out, and I've already done that and prepared it ready for you all to see. So this is a 10% sucrose solution here, and we'll be using this to extract the nematodes from the soil samples that we will pellet in those 50 mil centrifuge tubes. Okay, so I just ran through everything that we're going to do real quickly before we take them to the microscope and view whether or not we have nematodes or not in the samples that we extract. But I will be explaining each step of the process along the way as I do these steps. So the sucrose solution is ready. We can place it over here with the rest of the supplies and get ready to actually do the experiment. Okay, so with that said, just hold on as I prepare to collect the soil samples outside of this building. Okay?